Tonight is all about rivers, and our presenter, Vanessa Garayburu-Carusu. Vanessa will talk for about 40 minutes, and then we'll have time and space for questions and answers. If you have questions about the world underneath our rivers, I certainly do. Just by way of introduction, Vanessa is a scientist at Pacific Northwest National Laboratory, an organization she'll tell you a bit more about. She studies river ecosystems. So Vanessa is the chemistry team lead for the Wonders Project, which I'll let her define the acronym for you. But as I understand it, is really a global project to help us understand how rivers work beyond their role in just being water delivery systems to humans. So really studying the multi-dimensional systems of rivers. And I'm really excited to hear more about Vanessa's work and how the science is being. So without further ado.
Thank you. Let me start sharing my screen. There we go. So thanks again for the introduction. As I said, my name is Vanessa Garayburu-Carusu. I'm a scientist at Pacific Northwest National Lab. And today I'll be talking to you about the river sliver, exploring the hidden life and work of the Hyporheic Zone. But before we start with my talk today, I want to ask all of you a question. What is your favorite place in nature? And to answer this question, I want you guys to either use the link that I'm going to be putting in the chat, or grab your phone and with your phone, turn on the camera and scan the QR code. It's going to lead you to a website. And in the website, you're going to see this exact same question. What is your favorite place in nature? It's just going to be a one entry. It can be something broad, like, oh, I like the mountains, or I like the beach, or it can be something very specific like Mount Rainier or Glacier National Park. So I'll give you guys one more second to scan the code. And then here, hold on. We can see real life some of your answers. We had a lot of people that like the mountains, the forest. Ooh, some people like the tundra. Alpine lakes, streams and forests. The ocean, I think I'm right there with that person that likes the ocean. It seems that our numbers, one more submission. I'll see how our numbers are going over here. And this is our word cloud. And what it does is that however many people submit a certain answer, for example, forest, that would affect the size of the word in the word cloud. So over here, we can see that forest is probably one of the most liked plays among the people joining us tonight because it has a bigger font than everywhere, than other places. So I think our numbers have kind of leveled down. And so with that in mind, I want you guys to think, is your work or major directly or indirectly related to that place? And for this, we don't have to type it in the chat or we don't have to put it in a word cloud, but just think about it. And work can be something defined very loosely. It can be your day job. It can be the things that you volunteer for. It can be things that you're passionate about, just like anything that you consider work. So is your work or major directly or indirectly related to that favorite place in nature? And so just so you can learn a little bit about myself, my favorite place in nature is anything and everything with water. I hope some of you have gotten around this beautiful area that we live in and are able to recognize some of these pictures, famous Multnomah Falls, Palouse Falls. This is a picture of the Yakima River basin. These two are from the Columbia River in Portland. This is the ocean near Astoria. And the thing for me is that I just love water. And I feel very lucky that the things that I get to do for work are also related to this favorite place in nature that I have. And so, as you know, my name is Vanessa. And so I wanted to tell a little bit more about how I got here. I was born in Venezuela. It's a country in northern Latin America. And I did my undergrad there in chemical engineering. Then I moved to Albuquerque, New Mexico to do my master's in civil and environmental engineering with a focus on water resources. And then after that, I moved here to the Pacific Northwest to do an internship at Pacific Northwest National Lab that turned into a permanent position as a scientist working with rivers. And now, as I'm working as a full-time staff at PNNL, I'm also doing my PhD in Washington State University. So I mentioned PNNL, and I just wanted to tell you a little bit about this organization. This is mostly just language that I took from the website, but Pacific Northwest National Lab, it's a national laboratory that is located in Richland, Washington, so not very far from all of you guys in Yakima. And what it does is that it advances the frontiers of knowledge by taking on some of the world's greatest scientific challenges and technology challenges. The main core areas of research are related to biological and earth sciences, chemical and material science, computation and mathematical sciences, and engineering. And it does all of these while at the same time carrying towards enhancing our national security. So what do I do at Pacific Northwest National Lab? And my work is very diverse. I have done a lot of things through my time in research, starting doing a lot of lab experiments. Here you can see this is a picture from the experiments that I conducted during my master's, where I had columns packed with sediments from different rivers. Then I learned how to run a lot of instruments to try to analyze the data that I was getting from these experiments. I got to spend a lot of time in beautiful places, from like mountainous rivers in New Mexico all the way down to the Rio Grande and to other places here around our area that are very beautiful, including the Columbia River and the Yakima River. And I have also had the opportunity to work with other scientists to develop new instruments and new sensors that we can either put into the stream, like this one that we were building right here, or use in the lab to understand better how our rivers work. And then most of all, I've gotten the opportunity to interact with a broader community of scientists that are not only local to here, the Pacific Northwest, but also global and span different countries in the world. And I do that through this thing that we call WANDERS. So WANDERS stands for Worldwide Hydrobiogeochemistry Observational Network for Dynamic River Systems. And what this is just a consortium of researchers that want to understand how rivers work. And we come together, we design sampling campaigns that span different systems, and then we analyze the data together and just make inferences about all of these rivers that we study. So now let's dig into what the topic of this talk is about. What is the Hyporheic Zone? And I want you to think about a river. Just think about a river, you see the water, you see the trees, you see the area around. And imagine that you're walking towards the water. You're walking towards the water, and then all of a sudden you look down. You look down and you look at rocks, you look at sediments, you look at water. In this moment, you're standing on the Hyporheic Zone. So Hyporheic Zone, the word comes from the Greek, and it means hypo, below, and rios, flow. So it is the area below ground where there is flow. When we take a look at a zoom in cartoon version of the Hyporheic Zone, we can see here that is the area below ground where groundwater and surface waters mix. So in here you can see the different water sources identified by the different colors. Then if you zoom in even more, you can see that within those grains of sediments, there is life. There is active microbial communities that live there and they perform activities. And then within that water that flows through those microbes and sediments, there are nutrients, there are resources that these bacteria utilize to generate energy. And as they're doing these, they also remove contaminants and generate CO2. And so because of this function of removing contaminants, this area is also called the river's liver. So let's dig a little more deep on why do we study the Hyporheic Zone. So first, the Hyporheic Zone cycles energy, nutrients, and organic matter in our river networks. In addition to that, it helps to moderate temperature. And what this does is when during the summer, when the water is really hot, the circulation of water between the main channel where the water flows and the groundwater makes the overall temperature of the river water cooler. And then in the winter, it does the opposite effect. So it makes it a little bit warmer. In addition to that, it improves with water quality. Just like as we saw in the slide before, it helps filter out and remove contaminants from the river. It also produces greenhouse gases, specifically CO2. CO2, it's a product of bacteria respiration. Bacteria, just like humans, utilize organic materials, organic matter in the form of leaves or wood in the river to generate energy. And in the process of generating energy, they produce CO2. Moreover, they also provide habitat for macroinvertebrates and salmon eggs. So if we zoom in a little more, we can see this is a cartoon of the Hyporheic Zone. The different colors symbolize the water in the surface water and the water in the groundwater. And what we can see is when these two waters mix, we also create a very rich microbial community in the sediments because this mixes up whatever is coming from the water, in this case, the pink ones, from whatever was coming from the groundwater, which in this case is the brown. And so this is kind of like a super powerful microbial community within the Hyporheic Zone, and that it's capable of performing different chemical functions that are unique to just the water in the river or just the groundwater. And these functions are also fostered because of the optimal chemical conditions. So we talked a little bit about nutrients, we talked about organic carbon and how bacteria utilize these to generate energy. So it turns out that during this exchange of water between the two realms of the surface water and the groundwater, these microorganisms are always fed with the best of the best, which keeps them going and keeps their function for removing pollutants very active in the river. And then in addition to that, here we have a zoom in cartoon of salmon eggs. And what happens is that when salmon come and spawn in our rivers, they remove the first layer of gravel and then they put their eggs in the Hyporheic Zone. And when they do that, the eggs are kept protected so they don't like flush downstream when currents change. And they also serve as their home as they develop. So salmon larvae stays within the Hyporheic Zone until they are grown and strong enough to move in the river so they can avoid certain predators. And so here you can see a picture of that. So with all of that in mind, we know that the Hyporheic Zone is very important, but how do we study it? And what happens is that a lot of the processes in this Hyporheic Zone are very complex. And so in order to understand all of the different parts that make up this puzzle, we have to combine three key disciplines. And these are hydrology, biogeochemistry, and biology. And so if we zoom in at each of these disciplines, when we think about hydrology, hydrology is the science that focuses on studying how water moves. In particular for the Hyporheic Zone, it is how water moves in, out, or across the Hyporheic Zone. And in order to do this, hydrologists have to consider certain things. Among these certain things, there's the geology of the stream bed, Also the stream porosity, so how much empty space there is between the grains of sands or the gravel within the stream bed, the permeability, or the amount of water that is capable of penetrating through the subsurface at a specific time, and then the composition. And we can think about composition that water probably has an easier time to move through the stream bed if the stream bed is composed of things like gravel or sand. But if, on the other hand, the stream bed is composed of things like clay, it might have a little bit of a harder time. So hydrologists have to keep all of these conditions in mind. In addition, they also have to think about the river shape, the length, and the elevation, as well as the water velocity, because all of these things combined are able to tell how much water and for how long this water from the surface is able to go inside of the hyporheic zone and remain there, and how much time it's going to pass until it eventually comes back into the river. And so right now you can think as a hydrologist trying to understand how the water moves in the river across time and maybe across many different rivers can be very challenging, especially because it's hard to see like how every particle of water is moving in a stream. And so what they often do is that they rely on mathematical simulations that explain how the water moves. So these are equations that just say the water in the stream is supposed to move in this way. And with these equations, they're able to calculate how water gets transported and moved in our river systems and through the sediments. So that is the main role that hydrologists have within the hyporheic zone. Then we have biology. And with biology, we can answer the question of who lives there and what do they do. And so specifically, biologists use genetic tools to try to understand what kind of microorganisms, what kind of bacteria live in the sediments or in the form of biofilms. And biofilms are just a cluster of different bacteria that join together to the surface of sediment grains just so they can be more secure. And by being more secure, they're less likely to just flow away with the water when the water comes into the sediments. In addition to these, biologists can use genetic tools to see what kind of function would these bacteria have. And so are they capable of removing specific contaminants? Are they capable of doing a specific biological or chemical reaction? All of these biologists can help us answer. In addition to that, they can also help us look at what other types of organisms live in the stream bed, particularly invertebrates. And together with community ecology, we can look at micro and macro organisms and try to understand how they live together in the hyporheic zone. We can study the diversity of the microorganisms, meaning what kind of microbes or macro organisms live there. And then also trying to understand what are some of the characteristics that they have in common that it's only specific for the hyporheic zone. In addition to that, biology can also help us understand bioturbation, which is a phenomenon that happens when organisms burrow, ingest, and defecate the sediment grains within the hyporheic zone, which overall contribute to nutrient cycling and the biodiversity of the system. So lastly, we have biogeochemistry. As you can imagine from the name, biogeochemistry studies things that are a mix of biology, geology, and chemistry. And so for this particular thing, we call it biogeochemistry because it is studying biological reactions made by organisms that happen in the sediments within the hyporheic zone. So that's where the geology comes in. And so biogeochemists study the hyporheic zone in two ways. They either do it in the lab or they do it in the field. And their main role is to try to measure the overall activity of the hyporheic zone. And to do this, they need to track the concentration or abundance of specific chemical compounds that are very important for all of the reactions that the hyporheic zone carries out. Among these, we have things like oxygen and nutrients that are just key for bacteria to survive. By doing this tracking, They're also able to quantify how much contaminant removal or what are the process for contaminant removal within the hyporheic zone. One example that we can think about is the process of denitrification. This process is when we remove a contaminant like nitrate from the river, like bacteria are in charge of removing the nitrate from the river. And so nitrate is a contaminant that enters the river primarily due to agricultural runoff. And when there is too much nitrate in the river, there is a lot of algae growth. Nitrate is a nutrient. And when algae grows, some things can happen. Some algae are toxic. And when these algae grow, they produce toxins that are harmful for humans and animals. So we don't want that in our rivers. And then also, if the algae that grows is not toxic, they also lead to decrease in dissolved oxygen. And when there is no dissolved oxygen in the water, a lot of fish species and organisms cannot survive. So we rely on the hyporheic zone to remove this contaminant from the river. So biogeochemists study the natural capacity that different rivers have to remove nitrate naturally via denitrification. And so overall, this summarizes to biogeochemists study oxidation reduction chemical reactions in the hyporheic zone that lead to the removal of specific contaminants. And they can do these in the lab or in the field. So when we look at the hyporheic zone from the lens of a hydrologist, biologist, and biogeochemist, we can really appreciate all of the benefits that this area has to offer. So like we just reviewed, it has the potential to make reactions for nutrient happen really quickly. And so because nutrients in the hyporheic zone are consumed by bacteria, we can remove them from the river so they don't get to the ocean. If the rivers didn't have a hyporheic zone, nitrate coming from agricultural runoff will just end up completely in the ocean, creating dead zones in the oceans, not allowing for fish and other organisms to live and colonize in there. So this is a very important function for the hyporheic zone. In addition to that, it also helps remove other types of contaminants. We can think about things like trace organic compounds, which are not currently removed completely from wastewater treatment plants because their concentrations is really low. They come into our rivers because of industrial chemical runoff, pesticides, pharmaceuticals, and artificial sweeteners. And once they are in the river, the hyporheic zone has the capacity to absorb it, meaning that these trace compounds can get stuck into the sediment grains where the bacteria live, and then by getting stuck there, they don't get transported into the ocean. And they also have the capacity, some bacteria also have the capacity to transform these chemicals and remove them from the water. In addition to this, it can also filtrate and reduce the concentration of small particles. Among these particles, we can think about big sediment loads. It can help filter some of that, and microplastics. However, we have to keep in mind that if the hyporheic zone filters out too much sediments from the main river, then it can clog the hyporheic zone, not allowing for water to move freely in there and reducing their capabilities to remove other contaminants. And then lastly, as we mentioned, it can serve as a habitat for different salmon eggs, macroinvertebrates, and bacteria. And overall, it is a hub for biodiversity within our rivers. Now that we know a little bit about the hyporheic zone, I want to tell you some more about the things that we do within my project at PNNL and just like how we examine the hyporheic zone. So we care about understanding the processes that happen in the hyporheic zone at many different scales. And so we start by looking at basin scale impacts. And what this does is that it thinks about the hydrology, part of the puzzle. And within the hydrology part of the puzzle, it runs mathematical equations and simulations that are trying to understand at the basin scale what is happening in the hyporheic zone. Then we also look at the biogeochemistry in the hyporheic zone at a basin scale. And to do this, we conduct many different field experiments where we have sites distributed across all over the basin, and we looked at specific chemical reactions of interest. And then with that information, we can then understand better what is happening at the basin scale and tell that to the models. We also are interested in what's happening to the function of the hyporheic zone in the event of a disturbance. Specifically, we are interested in studying what happens when wildfires occur and how does that influence the water quality in our rivers and the overall function of the hyporheic zone. And then I mentioned that we look at things across scales. And so with these, we just go beyond one basin and we have multi-campaigns across different states within the continental US, where we have standard sampling and all of the collaborators select a site, sample, and together we understand how the hyporheic zone across each of the sites in the continental US is changing. So I want to take some moment in this talk to tell you a little bit more about our project in the Yakima River Basin. So we conducted a sampling campaign during the summer of 2022 with the goal of understanding how does hyporheic zone respiration change across the Yakima River Basin. And just to review, respiration is the process by which microbes or bacteria generate energy. And to do this, they need organic carbon, mostly coming from leaves, wood, dead cells, dead animals, all of that is organic carbon, and oxygen. And respiration is measured via oxygen. So our main goal was to understand the key drivers of hyporheic zone respiration and improve model predictions. Our motivation for this study is that the literature says that the hyporheic zone can contribute between 4 and 96% of the overall river respiration. From our perspective, it is really important to understand what makes a river contribute only 40% versus what makes a river contribute 96%. Understanding these contributions is going to help us, again, know what is the natural capacity that different rivers have to remove contaminants. This also is really important because this is just an equation of the respiration. So we have glucose as organic carbon, oxygen, and this process of respiration via bacteria respiration produces CO2. And so CO2 is a greenhouse gas, and we want to understand better what is the overall emission of CO2 across our basin. Another motivation for this study is that models that predict how this respiration is going to happen across different scales mostly include just the hydrology. So the physical properties of the stream, the length, the grain size, things like that, and do not really account for chemical or biological processes. So we think that if we deeper understand chemical and biological processes at a basin scale, we are going to be able to improve these model predictions. And so to do this, we use model predictions, which we can see here on the left, to select sites in the Yakima River Basin to test these predictions. So in this map, you can see the results from the model that uses only hydrology, so physical properties of the stream. And what we can see is that darker red colors are streams that respire more. Respiring more means that they produce more CO2 and are potentially more capable of removing contaminants. So when we look at the map, we can see that these streams are mostly located in the forest areas. And then as we move downstream, when we go into the more agricultural part of our basin and the more urban part of our basin, we see that these respiration rates slowly decrease. And so again, this is just a prediction, and part of our work is this true? How can we improve it? And so we selected 47 sites across the Yakima River Basin. You can see in yellow all of the different sites that we sample in the summer of 2022, and also in purple. The sites that we sampled that are in purple were sites that we sampled during the summer of 2022 and also more repeatedly since the beginning of 2021 to do a temporal study. And so I want to take a moment in here to acknowledge the Yakima Nation for being owners and caretakers of the land. And I also want to acknowledge the Yakima Nation fisheries for collaborating with us through this process and helping us collect samples and select sites and data usage that were according to their values and worldview. So with these sites in mind, we visited all of them, and now we can ask the question, how is observed respiration related to predicted respiration? But first, how do we measure respiration in the field? And the answer to this is via sensors. So this picture is a dissolved oxygen sensor. It's not very big. It's like probably the size of your cell phone, that big. And so we visited each of these sites and we deployed this oxygen sensor. And over here, you can see some of the different configurations that we use to put them in the rivers. In here, you can see this like blue thing. It's a wheel that cleans the window where the sensor measures. And the reason we did that is because we left the sensor in the water for 1 month. And during that time, you can imagine a lot of bacteria would be like, oh, this is a nice place where I can live, or I don't know, algae could grow. And so we didn't want that to interfere with the signal from our sensor. And so we have this little wiper that is cleaning the optical window of the sensor every now and then. And so these are some of the pictures of the way that we deploy these sensors. And these are some of the pictures of the sites that we visited. You can see that they're widely different. And so we try to pick a different range of land use conditions, vegetation, predicted respiration values, all with the goal of trying to understand Hyporheic Zone respiration. And so the data that we gather from these deployments looks like this. And what this is dissolved oxygen in the water. And you can see that it goes like down and up and down and up during the day. And this is because of the absence or availability of light. During the day, there is a lot of photosynthesis because there is light. And at night, we only have respiration happening. Respiration, which is the portion that we care about. And so via these sensors, we were able to estimate total stream respiration, but we want Hyporheic Zone respiration. And so to parse out the Hyporheic Zone respiration only, we also conducted water respiration measurements. And so to do this, we had a dark bottle that we submerged in the river. We filled it up with water. We put a dissolved oxygen sensor and we capped it. We kept it in the river for 90 minutes. We kept it in the river to control the temperature, so it was like the same temperature of the river water. And as you can imagine, there are microbes that live in the water. So there are probably less microbes in the water than live in the sediment, just because they like to live in the sediment in the form of biofilm. They feel very stable. But the ones in the water will also consume oxygen to generate energy. And so we estimate respiration as the consumption of dissolved oxygen over time. And so the data looks like this. And the rate at which they consume all of that oxygen is the slope of this line. And so now we have total respiration rate that we saw how we estimated in the previous slide, and water respiration rates from these little bottles. If we subtract one minus the other, we can get Hyporheic Zone respiration. So we completed this experiment in the summer of 2022, like I mentioned, and we're still processing that data. So I don't have fully final results to show you from this field campaign, but I have complementary results that we can talk about and are going to help us get to our main goal while we're processing this data. And so our complementary measurements relate to how do we measure respiration rates in the lab. And so the way that we do this is that we collect sediments from each of these 47 field sites and we bring them into the lab. Then we put them in a vial like you see here. So these are sediments from the river, and then this is water from the river. We cap it, and then we keep it at shaking at constant temperature for 2 hours. And we measure dissolved oxygen concentration. And the way that we do this is we use fiber optic technology, which you can see here in this picture, and we use this little pink dot. So this little pink dot actually has chemistry inside it that permeates dissolved oxygen in it. And when we light up this pink dot with one of these fiber optic cables, there is an excitation emission signal that is sent into the dot, and then we can get the chemistry of the dissolved oxygen without having to open the water, taking a measurement, nothing like that. Just we don't have to touch the vial at all, and then we can see like we get data that is very similar to those bottles that we incubated in the river. And so we have dissolved oxygen and how it is consumed over time. Then the respiration rate is the slope of that plot. So with this, now we can go back to our question and ask, how is observed respiration rate related to predicted respiration? And over here on the left, I have a map of all of these laboratory results of Hyporheic Zone respiration rates. And so the respiration rates are presented in different colors across all of the different sampling sites. The lighter colors are rivers where the Hyporheic Zone wasn't very active. And as the colors get darker and darker, those symbolize rivers that there was more activity. And one thing that we can see over here that I have circled for you in green is that there were 2 sites in particular within the whole Yakima River Basin that were very active compared to all of the rest. And so when we see something like this, we call these regions like hotspots because they're just hotspots for biological activity, in this case, Hyporheic Zone respiration. So going back to our question, how is this related to the predicted respiration rates? And so I put here again the plot that we have already seen of the respiration rates. And just to review, red colors are higher respiration and blue colors are lower respiration. The model says that we're going to see higher respiration in rivers that are in the forest. But as we move down, we're going to see less and less respiration as we go into the agricultural and urban areas of our basin. And so the main thing that we can notice when we look at these 2 maps is that the map of the lab results has a lot of more variability in terms of what sites have high versus low respiration, while the model just seems to be a gradient that it's decreasing as we move downstream. The other thing that we can observe is like I drew a green circle here, roughly around the same area where these purple sites are. And when we look at the model predictions, we see that there is no hotspot. We don't see that there is something specifically different about this area that is going to make a river be more active or have higher Hyporheic Zone respiration. And so what this is telling us is that there is something really missing from the model and that we have to find out more to see how we can complement these predictions and make them more accurate and potentially be able to predict these hotspots. So now you might all be wondering, what makes these sites so special? And the answer for this question is still ongoing. I mentioned earlier that we took a lot of different kinds of samples. So some of the things that we're analyzing are general water chemistry. We also collected sediment samples. Here's a picture of how we're seeing sediments from the river. And we collected data from like water samples to analyze in depth all of the different organic carbon, Maybe it was just that they didn't have enough organic carbon. And because organic carbon is part of that respiration equation, they just couldn't respire as much as they wanted. We also collected pictures for grain size distribution, just so we could understand better how the water in the it's moving through the sediments and how the microbes are attaching to those sediments. And we collected something that we call, it's a cotton strip, and cotton is just 100% carbon. So it is what bacteria like. And so we deployed cotton strips. You can see in here, all dirty, but it's just a piece of cotton that it's coming from a canvas for a painting. And we put it in the river for 1 month, and we just let it be and said, okay, microbes, eat it. Right? And then we collected those data as well as another way to try to understand how microorganisms are capable of respiring across our basin. So soon enough, we're going to have all of these data, and we're going to be able to tell what is happening across our basin, and specifically across these 2 sites that seem to be very active. So together, all of the different things that I talked to you about bring us back to the science cycle. And so what the science cycle is that we initially had a model that made simulations of how respiration rates in the Hyporheic Zone would change across the basin. And then we said, okay, let's conduct field work, and let's grab field measurements along the basin to test the hypothesis. The hypothesis is we can improve these model observations if we understand better the biology and the chemistry of our rivers. Now, when we work on testing this hypothesis, we are able to utilize the information that we gain, in this case, physical, chemical, and biological information from our basin, and put it back into our models and generate new predictions that are going to help us create new fieldwork, to take new measurements, and keep this cycle going until we are able to understand with enough precision what is happening in our basin, to the point that this model is able to tell what is happening. And we really need to work on this, because this is the tool that we as humans utilize to understand how our basin is going to react under changing climates. And this is the tool that is going to help us act more quickly in the event of a disturbance, like in wildfires. How can we keep the overall health of our rivers when something happens? So we need to understand what's happening now and make tools that are going to help us predict what would happen in the future, so we can respond quicker. And with that, I want to thank you all for listening to this presentation. And I want to thank our sponsors, the Environmental System Science Biological Division of the Department of Energy, and all of our team. Fieldwork like this, or science at the scale that we do at the National Labs, wouldn't be possible if we didn't have a great team to work with. So with that, I would like to open it up for questions.